to see the chemistry and biology at the same time. So Lars, please take over the stage, and looking forward to your lecture.
Yeah, thank you, Yi-Wong, and thanks for inviting me to give this presentation. I like it that I was after Matthias. Thank you, Matthias, for the presentation. You gave a good introduction, and I learned something about chemistry. I'm not a chemist, Yi-Wong. You mentioned I did chemistry in Wageningen, but I have to disappoint you because it's more environmental science and environmental biotechnology that I work on. So today I'd like to talk about our process of CO2 to protein system. That's a two-stage system, and I want to first acknowledge the co-authors. One of the things that is great about CORC is that I can do things that I can't do in just my little lab in Germany. So we have obviously a center where there's much more possibilities of doing work that can leave your lab. And so I want to thank Alfred Spoorman, Lars Angenent. He's known as Lars O. I'm Lars A, obviously. Michael Kofod, Martin M.B. Jensen, Thomas Lundgaard, Torben Larsen, Juan Ramirez, and Lisa Schmitz. I just wanted to acknowledge them because without the larger team, there's not much really that you can do. And I think also Matthias referred to that part of his institute, that he can build pilots and stuff, and that you need a bigger organization for that than just having a small lab in a university. So I really appreciate that. It's also a very good time today. I was actually a little bit busy because yesterday there was a big announcement that was done by the Bill and Melinda Gates Foundation together with the Novo Nordisk Foundation that have supported research in this specific area of doing carbon bonding to acetate. So just referring that to Matthias' talk, and then converting that acetate into, for instance, protein, but you can also make other chemicals from that. And that's exactly what the talk is about today. So I want to also thank the founding foundations that have made this possible. So it's the Humboldt Foundation, Sprint. I'll talk a little bit about it, the CO2 Center itself, Bill and Melinda Gates Foundation, or now Global, and the Novo Nordisk Fund, obviously. So I just wanted to make sure that we can only do something like that is so huge if we work with many people together. And this is actually my first project that would I say that, okay, we can really scale up by working with different foundations. So this start was made with money that was left over in Humboldt professorship, in which I was able to support 2 postdocs, Lisa and Juan. And then we obtained a grant from Sprint D in Germany. This is an organization that brings technology from the lab out in the real world. And we got 200,000 euros to do a techno-economic analysis. This is a person, Andres Lisset, in my lab, plus consultants that I will mention later. Then there was the grant from the CORC itself, because they have also a granting function. And there we were able to achieve and to be awarded 2.5 million euros to actually scale up our process. So what we've had in the last was 2 liters. Now we're scaling it up to 200 liters, for instance. So that's obviously very important to be able to do that, not only to receive the money, but also to find a place where this is housed. And we do that in Falun, which has a very close connection to CORC. And this is also why Lars Angenent, for instance, is mentioned here as the co-authors. Then there was a grant from the Office of Naval Research, or now Global. They have a seat in London. They're interested in food production as well, and they awarded us with $500,000 to fund 2 postdocs that can work together with the pilot plant. So the pilot plant itself doesn't run itself, right? So we have some money to do that. And then finally, yesterday, we heard that there is money coming from the Novo Nordisk Foundation and the Bill and Melinda Gates Foundation, and to just Alfred and myself awarded of 1.6 million. Because what's really important here is that we still need basic research to make this process more efficient. So that is done supporting 5 postdocs in a management function to really do basic science on our microbiology to make it work much more efficiently and higher kinetics. Okay, so the problem that we are facing is immense, and we've already heard from Matthias that Denmark doesn't look so good. Denmark is by, I don't know, Matthias, I forgot it, by May or even earlier, basically they already used up all their resources that they could basically replace in 1 year. And I think part of that is actually due to agriculture. And I'm not sure what the real reason is, but I would predict that, and this is a thing that both in Denmark, and I know a little bit more about the Netherlands, they have a big industry of agriculture industry that they have, for instance, many pigs, more pigs than people probably, and they import animal feed, and this is protein that often comes from Brazil. And I know for the Netherlands that that the size to grow the protein that's used in the Netherlands for the pigs and other animals is 7 times the surface area of the Netherlands itself. The Netherlands is very small. So you can immediately see that the Netherlands or Denmark would very quickly run out of resources. This is not the only problem I think that current agriculture has, but we have obviously a situation with greenhouse gases, CO2 production, but also methane. And I think in this thing that I found on the web is that 26 of the greenhouse gas emissions come from food production. And that's just one problem that we face. We have so many things. We have water issues, eutrophication issues through conventional agriculture. And this is partly, I think, because agriculture is not very efficient. But I can also think about other problems, like we have a lack of insects, so the insect diversity went down and the amount of insects went down. We have a diversity of almost everything. Wildlife in for instance, the Netherlands and Denmark. We have groundwater problems, and the list is actually very long. Now, instead of blaming the farmers for all these problems, it is our problem. It is our cause, because what we wanted as a society was cheap food, and we wanted to work as efficiently by using basically fossil fuels. So I do not want to stand here and blame the farmers. I think that we have to work together with the farmers, and also they can be the solution of the problem. So I see a problem in conventional agriculture, but I don't blame the individual farmer for that. I blame our societies and the push for cheap food and super efficient systems. And I think that what we want to do is provide technology that can help solving some of these issues. So the goals of that, so obviously what I see as my own goal as a technology developer is that we want to develop a sustainable and economical biotechnology production platform to produce pure protein. This would be then used as directly as human food, not as animal feed, but as human food from CO2. In the meantime, obviously, why you need many people to work on that, because the scale of the first goal would be very large, is that we want to considerably reduce the CO2 emissions from protein production. We want to thereby also reduce the environmental footprint of human food production. And for me, what's important is to develop a sustainable agricultural sector where farmers are stewards for the environment and the countryside, and that the farmers have a sustainable income. So you cannot just yank all the animals away and say, now it's over. No, you need to provide, we need to have a transition period where we provide farmers with a real income and provide them with technology which we are developing, so that we can build together a sustainable food production system. Because we do need vegetables, fruits, nuts, plant proteins, grains from farmers, and we need to do it with biodynamic methods. We cannot provide food in the way that we've done it, because we saw all the problems that this has caused. So I want to make very sure that the technology that we develop is in no means replacing farmers. No, actually, I want to strengthen the farmers with it. And obviously, we want to also provide local food production. So I don't think it's sustainable to ship or fly food around the world. And I think that we need to provide local food production. So if this is in Denmark, this is a Danish case, then we need to strengthen agriculture in Denmark in a sustainable way. But also in the developing countries, obviously, we can provide food production. And so these systems are going to look a little bit different depending on which regional area you're going to be in. I just want to make sure that we make that point that the farmers are not the cause of the problem. It's us who are the cause of the problems, and it's also us who have to solve this. Okay, in terms of like the technology that we're interested in making protein, single cell protein. Another word for that could be microbial protein. This is nothing new. So in the 60s and 70s, we had other crises, and this came out of that crisis. And this is an example of actually a successful example of a company in the UK that had originally started to make single cell protein for animal feed. That didn't work out, and another company called Marlow Foods took over the process, and you see an old picture here in the UK countryside, and they developed an edible protein, this single cell protein, or microbial protein that they call corn, and you can buy this in the supermarket in the frozen section. Maybe you have seen this before. I've eaten it myself. I like it actually. And it's a fungal strain that they worked on themselves. They isolated themselves, and they got regulatory approval to use it. And it's called Fusarium venenatum. It's a single cell protein. It has certain characteristics that it gives you a good bite when you eat this protein. Obviously, here is the reactor in the middle. You can see that here of the system. I think they added another 3 of these reactors. It takes a lot to go from a reactor into something that we want to eat, right? And there's a lot of development by food scientists necessary to make this into a product that is cheap, relatively cheap, or at least cheaper from the protein from animals that we get, and also that is something that we want to eat. So there is a big, and there's a lot of people in the world working on that right now, on alternative proteins. Okay, so we have conventional agriculture, which we know, and then the middle is the single cell protein. That's the example that I just gave you. So let's say we start with glucose in that sense. By the way, I forgot to mention it. The input of there is glucose. So this glucose is still coming from conventional agriculture. It's solving the problem somewhat, but we still have problems with growing all that glucose. So what we want to do is we want to use CO2 directly, and in terms of the protein production, take conventional agriculture out of protein production. It doesn't mean we want to get rid of agriculture altogether, as I mentioned before, because there's many other things that we need, including probably proteins and other food materials that you want to mix in with the protein that we're producing to deliver a product that we actually want to buy and want to eat. This is very important. In terms of the process that we envision here is that, and coming back almost similar to what Matthias already just showed us in the previous lecture, is that we also want to use hydrogen, and we want to use electrolysis to split water with renewables. And he is luckily in northern Germany. I'm unlucky because I'm in southern Germany, but we have a lot of CO2 though, Matthias, so maybe we should talk. We split water into hydrogen gas and oxygen. And then what we want to do to make the carbon bonds that Matthias was talking about is we do that biologically in a first stage. And these are anaerobic bacteria that we call acetogens, and they produce acetate. So it's a carbon-carbon bond made from CO2 and hydrogen by acetogens. And then we pump the acetate into a second bioreactor where we will grow the single cell or the microbial protein that I just talked about. So now acetate is the electron donor, and oxygen, which is coming from the electrolyzer. Most people don't use the oxygen from the electrolyzer. We want to use that oxygen from the electrolyzer because it makes our process more efficient, biological process more efficient. And here we produce protein. These are just the yeast cells as one example, or the venenatum cells that we just saw in the previous example, which has about 50% protein. So the product, in a sense, is the dried cells that we produce. Obviously, there's different levels of post-treatment that you can do. You can extract the protein, or you can keep it together with some of the material but that's maybe for another time we can discuss that. In the end of the day, we will have biomass that's coming out, and often there is also nutrients in there, so it has some nutritional aspects that food scientists would have to determine. So this is our process. The CO2 would come from industrial off-gases, but could be any source. And part of the circular economy, we would also like to see how we can recover and recirculate nitrogen and phosphate. And this could come from wastewater. There are safe ways to do that. Of course, you have to be careful in communicating that to the people who will eventually buy the food. But this is possible. We calculate theoretically that if we produce biomass this way, so it's just the biomass, which is about 50% protein, that the energy input is about 24.4 milligrams. We produce 24.4 milligrams of biomass per amount of kilojoules that we add. And this is coming back in a comparison. That doesn't mean much to us, but I just want to compare that into the next slide. Because you could make biomass from CO2 in different ways. It's not just our process. We have this two-stage process, but you could potentially do it in one stage. This is with knallgas bacteria. They use a different pathway, and they basically, you mix, if you do it in one stage, you would have to mix the hydrogen, the CO2, and the oxygen together for this reaction. That works. There are companies like Solar Foods in Finland that do this process. We calculate that you get about 9.5 milligrams of biomass per kilojoule. The protein content may be a little bit higher there. In our process that I just showed you, where we have two biological processes or reactors in series, it would be about 24.4 milligrams per kilojoule. That's already shown. If you would do that electrochemically, for instance, if you make acetate electrochemically, you could do a little bit better. And so then the electrolyzer and the first biological process would be replaced by an electrochemical system that makes acetate. You do a little bit better energetically. There may be advantages to doing that. So, and this is also why what the Bill and Melinda Gates and the Novo Nordisk Foundation is funding, to look at, okay, where is the acetate going to come from to make this protein? Should it come from biological systems, like that's the process that Alfred and I and Lars and others are working on, or does it going to come from electrochemical systems, or even from chemical systems? Okay, so we like to use acetate, but you could also use intermediates like methanol or methane. You get the numbers are given here. We have chosen for acetate as the intermediate because we can convert that, for instance, into yeast. So that's kind of a comparison. So we are very happy with this, using acetate as an intermediate and to use this two-stage system. And in very simple drawing, it looks like this, obviously, that you have in the first reactor. You could run that at 35 degrees Celsius or 65 degrees Celsius. You pump in hydrogen and CO2. It's an anaerobic first process, and you have to feed trace elements. Obviously, this is a biological system. And we try to minimize the amount of vitamins and nutrients that we'll have to add, obviously. In the second stage, we said acetate was being pumped in the second stage. Here we put the oxygen from the electrolyzer in. We produce the biomass, which is about 50% protein. One of the things that I want to tell you is this mineralization process that happens in the second reactor frees about 50 to 60% of the carbon bonds that we just made as CO2 again. So this is obviously not very exciting, but it's a thermodynamic reason for that. It's not that we're going to stop that. But in our process, what we want to do, since we have a two-stage system, we could and we will recycle that CO2 back into the first stage. So that if you put a boundary around the total system, we won't release that CO2 again. We have started the process. We published a paper on this, and we've used Clostridium undalia, which is a cetogen, this anaerobic bacterium that can make acetate, and we have published a paper on there. We are also working with a thermophile currently, and which is called Thermoanaerobacter kiewii, and we are comparing the two different cetogens. In a sense, we want to develop the hardware for the process so that we can change the microbe if we want to, but we're also doing basic science on optimizing what we believe is a strain that has industrial perspectives. Now, in the second stage, we have used just baker's yeast. So Saccharomyces cerevisiae, about 30 degrees, running that to make the protein. This can also change. So in our process, in the hardware, the reactors, we could grow something else and we could change over time, or we can grow more than one organism in different reactors and to combine it into a food. So this is all possible. So why have we started with yeast? We've started with yeast because this is already sold in vegan restaurants. This is a vegan restaurant in Aarhus that I visited a year ago or something like that, and they had a nice pasta dish, and you can see on top of the vegan the yeast flakes on top of this. Was a vegan meal that they sell there, and I went to the kitchen. I said, where did you get your yeast from? And here see a picture and they bought it just in a normal supermarket next door. So this is not something that it's. And so this is what we want to produce. So yeast could be one example of something that people would already eat. It is an acceptable product and not too far off. So this first trial with Clostridium dahlia that we did and that we published about, we achieved high acetate production rates. This is in the range of 1 gram of acetate per liter reactor volume per hour. So this is per hour. This is a level. This is an industrial level. This is what the DOE, Department of Energy in the US, wants to see. So this is industrial. Of course, when you optimize it further, you'll get much higher level when you're a company and we're doing the basic science to really understand we can increase that further. The protein production in phase B was too low, I would say. This also needs to be improved, 0.07 gram per liter per hour, so a lot more work is necessary there. The acetate carbon yield, so the carbon that went from CO2 to acetate in the first stage is about 82%, so that's not bad. But then, of course, we said that that some of this, like maybe half or 2/3 of this carbon, is released back as CO2. So the carbon yields for biomass in stage B is not so high. Overall, the combined, we had 25% because we did not recycle that CO2 back to the first stage from the second stage. So this is obviously something we would have to work on to make that a much higher overall biomass carbon yields from CO2 into our product. Recent studies in the lab. So in our lab, we run a lot of bioreactors, and we would do both microbiology, but also bioreactors. I'll give you some examples now of the bioreactor work that's done by Lisa Schmitz, who's a postdoc in our lab, as I mentioned earlier. So it's a 2-stage process. And you have stage 1 where we make acetate, stage 2 we make the yeast. You can see here on the right, you can see a small picture. We have the reactor here to make acetate on the left, the reactor on the right to make yeast. And you can see it's pretty much like a lot of self-built stuff. It's 2 liters volume for each of the reactors, a total of 4 liters total volume. And you see here's the product. So this was at our highest production rate. The yeast turned a little orange, and I'm from the Netherlands. And our national call is orange, so that made me very happy. But another scientist told me that maybe there's some stress involved, and that's why it turned orange. So there's a lot of research that needs to be done, as you can imagine, to really kind of scale that up. So recent studies in our lab with this thermophilic acetogen shown that we are doing better. We are now, in the beginning, it wasn't, we get 0.3, then over time you see here, this is a moving average in orange. Again, orange, my favorite color. You see here 0.6 grams per liter per hour. That's a thermophilic acetogen. We were not doing as well as the mesophilic acetogen. That was the C-Lumda that I told you did 1 gram per liter per hour. But we for sure will get there. This is one of the first times that we're really using this microbe in kind of, long-term continuous operating system. In terms of the yeast, and so the acetate from that first stage going to the second stage, we find that the gram dry, this is cell dry weight per liter per hour. I quoted you about 0.07, I think. We're doing better than that. So we're like now often reach 0.15. So we have doubled that, but we need to further increase that, and we're working on that. So what we are working on, not only in my lab, but also at CORC, is obviously, and I want to mention is that I'm at the University of Tübingen, and that I've put in the CO2 Center and the University of Aarhus, because I'm also have a function in Aarhus, 50% professor in Aarhus. We will be scaling up our process 50 times. So we go from 4 total liters to 200 liters. We will have to increase that production rate by a combination of different types of research. We would like to recycle the CO2 from the second stage, aerobic stage, to the first stage, and how to do that best. We are working on that as part of the scale up. We want to choose wisely in terms of what type of single cell fungi or yeast we actually want to operate, or if there are several ones in our systems. And what's very important, obviously, is the nutritional aspects of our production system. And this is also in collaboration with Aarhus food scientists. And this is obviously super important. In the end of the day, you will find a product that is a mixture of different things, and they have to provide the nutrition that we need, not just the protein. Just with protein, you will not be able to live very long. So the scale up in Follum is done, completed on paper. The design is being done on paper, and now it's the question of getting quotes and having it built. This is a process flow diagram that Juan Ramirez from my lab has developed. And you can see that when you scale things up, it becomes a little bit more complicated, which is good, right? We're engineers, so we like complication. Why in Follum? Well, first of all, Follum is connected to CORC. They have a lot of very good scientists and engineers working there. We're working with several of them, including Thomas Longdaard, as I mentioned before. The other thing that they have is they have the facilities, they have the buildings to house the reactors, but they also, what they have full-scale anaerobic digestion. So they make biogas from a mixture of plant material and animal waste, and we need CO2. And we don't really care where the CO2 is coming from, but this is one example here. This is his first stage thermophilic anaerobic digestion, and the second stage anaerobic digestion with a ball you see here where the gas is being stored. So plenty of CO2, because biogas has about 40 to 50% of the gas itself is composed of CO2. So you can either then first separate the CO2, put the methane into the natural gas grid, the CO2 into our process, or you can even take the biogas together with the 50% or 60% methane and just feed it into our first stage. That's also possible. So we will use real gas for our pilot plant. That's also important. We are working also on a techno-economic analysis. You have to kind of keep in mind always that this has to eventually lead to an economical process. So you cannot do a techno-economic analysis early enough, see what are the sensitive parameters. And if you look at the operating expenditures or OPEX, you can, for instance, look at what's the cost of CO2. Let's say that I have to buy CO2, because of course I need a good quality CO2 always at my production facility. So let's say that will cost $300 per ton. Maybe there's some credits that I get later back, but this is the number that we set. Or we said, well, we have to buy electric power because we produce hydrogen. So what is the price that we can buy this at? Let's say 13 cents per kilowatt hour. That's a little bit on the low end, but maybe we'll have more renewable power in the future, and this is a realistic price. If you do then the comparison of the operating expenditures, we see that CO2 under those conditions would take 7% of the operating cost. The media would be 2%, so it's not that sensitive. The nitrogen would be 1%. This would be something that we buy rather than recycling it from the wastewater that I talked about before. But the electricity has a very big impact on operating expenditure, because we have to produce a lot of hydrogen and oxygen through water splitting, and that's an expensive process. So you can immediately see that what we have to look for and where we can save costs is on the energetic side. It's still an energetic process, maybe less energetic than conventional agriculture, but it is still an energy demanding process. So this was done with a consulting firm called Connectomix. I've learned now that it's very hard to do TEA by yourself. So we have teamed up with Connectomix through the Sprint D process. And they're developing an Excel-based techno-economic analysis that we can use where we can import data all the time, that we have new data, including things that are coming that we're learning from the pilot plant. The post-processing is also super, super important, and we will work with food scientists in Aarhus to look at that. You have to do several things. You have to do autolysis, autolysis, because the RNA levels will be too high in the yeast, so you have to heat it up, and that will destroy the RNA, some of the nucleic acids. This will also be a loss of some protein. There needs to be ultrafiltration and maybe a spray dryer, because you need to deliver a product that you can actually sell. So this post-process is something that I personally know very little about, but Aarhus luckily has people in the food sciences that could help us with that process. I think what's also important to mention to you is that okay, what we have invented or what we found out, in a sense, is something new, and it's a new combination of different steps, but each of these steps has been scaled up. So if you want to do this, like if the Gates Foundation with the Novo Nordisk Foundation is serious about this, and we are successful to develop the basic science and the basic science around it, then they could scale this up very quickly, because we have full-scale and industrial-scale electrolyzers, right? This is one picture from John Cockerell, but there's other companies. We have a company in Chicago that does gas fermentation. They're using almost the same strain to make ethanol. We want to make acetate, but the reactors have been scaled up, and they have reached very high production rates. This is a company called LanzaTech. This is a picture of at a steel mill where the carbon monoxide is being used in the system like this. So the hardware is done. And then I talked about Marlow Foods. This is a more recent picture where you can see two of their big reactors, very tall reactors here, that does the phase B. So we have basically electrolysis. Phase A is being scaled up, phase B. But of course, that's the hardware. And we're doing science on the software. The software are the microbes. And of course, this integration, how to integrate all of this and to make it a more efficient process, is something that we still have to do a lot of research. And that's why I'm so excited to be part of CORC, to make this a reality. These are the people I want to thank from my own lab here in Tübingen. Just thank you for all the funding and the people who support me with the research. So that's basically my